Okay, very good afternoon to everyone. I thank the organizers for this opportunity. I would like to give an overview of some of the work that we have done on this amyloid beta aggregation and phase separation. Um, the liquid-liquid phase separation is not a new thing. It's been reported, studied, explained a long time ago in the context of globular proteins and colloidal systems. What you see here is a classical, a lysozyme protein, which is a type of a globular protein. And people have experimentally measured what happens when you create a protein solution of different concentration at different temperature. You kind of create this kind of a liquid-liquid phase separation. It basically means that two liquids phase separate, and if you measure the concentration of protein in these two, they basically have different concentration of the protein. So this is just an example, but there are a lot of other examples one can cite. And initial interest on this system were to understand what are the requirements to see liquid-liquid phase separation, more from a colloidal standpoint. So people are actually interested in looking at some models, interparticle interaction models that could predict this kind of liquid-liquid phase separation, or LLPS in short. What you see on the left-hand side is phase diagram predicted for a Lennon-Jones potential. What you see here is the Lennon-Jones potential between a pair of colloids. And so here we have temperature in reduced units, and here we have density in reduced units. And you see the classical, vapor-liquid equilibrium, and there is a solid-liquid equilibrium, and then there is a vapor-solid equilibrium. Okay, it's very typical of any molecular fluids. On the other hand, if you look at this system, so here this Lennon-Jones potential was taken, but then it was shrunk in terms of the range of the attraction. So the difference between this and this is that you can see the range of attraction kind of cuts off around about 1.4 or 1.3, but here the interaction goes all the way up to some close to 2 times the diameter of the particle itself. So if you make the attraction between a pair of colloids short-ranged, then one can actually predict this kind of a phase separation. You can see that there is a fluid-solid coexistence above, but then this what we know as vapor-liquid equilibrium basically gets buried inside the solid-fluid phase boundary. So this is something one can calculate with this kind of a model. And then further on, people actually made a generalization saying that if the ranges of attraction is less than 25% of the particle size, then you might see an LLPS. Okay, so that was a kind of a generalization. So it can be protein, it can be particles, it can be any rigid object with no internal degrees of freedom. People actually show that this is one possibility. And subsequently, there are also other interest in looking at non-isotropic interactions. Okay, the previous model was an isotropic interaction. So what I mean by isotropic interaction is that the nature of interaction depends only on the separation distance and not on the orientation. Okay, so that's isotropic spherically. But then there's other development that happened where particles were actually shown to have some sort of attractive patches. With regard to protein, for example, we know that there are some hydrophobic patches, there are some sort of charges on certain surfaces. So when you look at the interparticle interactions, one can think of beyond the isotropic interaction. So that's where the patchy interaction was brought in. And people showed that even in this kind of a patchy interaction, one can also predict this kind of phase separation, which is buried below the fluid-solid interactions. So these are the things that have been already done for quite some time. And one more thing you can also say is that from the colloids point of system, there are other things like there is this a gel through percolation, and there is a glass formation, which is also kind of intersects or interferes with the liquid-liquid phase separation. So there's a lot of work being done on this particular field. I'll come back to this slide a little later. For example, here, what you see on the left-hand side is an experimental work on lysozyme salt conditions. So again, you see this kind of a binodal, the liquid-liquid phase separation on the top. But then when you come deep, you see there is a glass line that actually connects or intercuts the LLPS itself, or the liquid-liquid phase separation. In terms of cartoon, it is shown nicely here that you have this, the red line that you see, that is the liquid-liquid phase separation boundary. But then before it terminates, you see there is a glass line that kind of kind of cuts. So suppose if you bring a system from here to here, you are more likely to get arrested gel or arrested glass even before the phase separation happens. So the point I'm trying to make here is this LLPS is not only just an equilibrium phase separation. If you quench deep enough, you can even interfere with the glass line and the percolation line and so on. So the dynamics of liquid-liquid phase separation also is very important to take into account. Okay, on one hand, we can predict LLPS from an equilibrium point of view, but from an experimental point of view, there are a lot of kinetic effects that brings in additional features, additional challenges. So this is an example where the attractive glass lies close to or within the unstable region. So what I mean here is that this black line that you see, that is an attractive glass regime, and that happens to be below the LLPS. This again is from the context of colloids or globular proteins. And I can show more such examples with more intricacies, where in this case, for example, we have a dynamical arrest happening deep inside the LLPS boundary, or you can have this kind of, like I said before, you can have a glass boundary intersecting the binodal. A lot of experimental data that you can see on globular proteins. Now, even though this LLPS was very well known, but I think the recent, recent interest or renewed interest on LLPS happens because of this, relevance to biological systems. Many of you might have actually seen this kind of picture. This basically shows a kind of a cartoon of a cell in which there are many objects which are actually membraneless. Okay, so there are many condensates or droplets which does not confine through a membrane. Okay, so generally we think cell means there is a membrane bounding it, but there are many objects that are found. You see there's a P body, U body. There are so many nomenclature that is out there. All this spherical thing that you see, these are all membraneless condensate, and that's when this nomenclature biocondensate or membraneless organelle came in. So ever since this topic was brought in, then there's a lot of interest on studying liquid-liquid phase separation in macro biomolecules like proteins, RNA, DNA. I think we already saw many such examples in this conference. So now I wanted to kind of now switch to what our interests are. So we were actually interested in studying this amyloid beta, which is a kind of an intrinsically disordered protein. And I will show you towards the end of the lecture that this is kind of a protein also shows a liquid-liquid phase separation due to some of its intrinsic dynamical characteristics. So to kind of tell you about what this amyloid beta means, there is a amyloid precursor protein APP which has about around 770 number of monomeric units, amino acids, residues, and that's supposed to form some functional role in our body. But this protein also, it's actually a membrane-bound protein. Okay, so what you see here is a membrane, and this big stick that you see here, that's N-terminal, C-terminal, this entire protein is APP. And these proteins are actually subjected to enzymatic cleavages through various enzymes. And again, these cleavages are required for some normal biological functions. But once in a while, there is a mistake that happens in the cleavage point, and that leads to a formation of an amyloid beta monomer, and that seems to have some harmful effect. For example, in this case, if you consider on the left hand side, this is supposed to be the right enzymatic cleavage. So you have this APP here, one particular enzyme basically cleaves at this point, and this is released, and there is another enzyme that cleaves at this point, and this gets released. So this is a normal bio process. On the other hand, sometimes if the cleavage is incorrect, for example, instead of cutting here, if it cuts around this point, then it basically leaves this the green ones, and those are kind of called amyloid beta monomer, and they tend to aggregate. So purely from a biophysical point of view, this is just a mistake of an enzyme cleavage process. So when this, if you look at these segments, typically you might have 40 residues or 42 residues. Sometimes you get 36, 38 also. Okay, so there's a range of sizes one can create. So we are saying that from a 770 sized protein, we make smaller segments which are predominantly hydrophobic, but they also have some charge residues. Later on, I will show you that. And we are interested in studying what happens to these amyloid beta proteins or monomers. So what happens is that once those are cleaved incorrectly, and those proteins tend to aggregate, okay, they form aggregates, and then once they get aggregated, also form some sort of a plaque or network that kind of deposits in important tissues like brain tissue, neurons, and so on. And it kind of affects the neuro signal transmissions and transductions. this is a cartoon picture. This is supposed to be like a normal neuron in the brain. And then when you have this kind of an aggregate getting deposited in this tissue, it kind of interferes how those neurons will communicate. So that leads to a lot of cognitive disabilities, Alzheimer, Parkinson, many other neurodegenerative diseases. So from a medical point of view, people actually look at this problem from a PET scan kind of methodology. So what you see here is a normal brain, and this is a Alzheimer diseased patient's brain. And what you see is you see a lot of this red spot that basically says there's a presence of many of these aggregates. Okay, so they put a dye, and the dye actually specifically binds to these plaques, and that's when you see this red signal here. Okay, and so this Alzheimer is something that one can check through a PET scan and many other imaging techniques. So these are the biological significance of these proteins. Now there are a lot of studies people have done on looking at how these amyloid beta aggregates in vitro conditions, meaning you take this in a glass tube or a test tube and then study how they aggregate and how can we understand the kinetics and what determines those kinetics and so on. So one of the very simplest thing you can do for people like me who are not really an experimentalist but want to get into an experiment, what you do is you do something called this fluorescence assay. It is basically you put a dye, this is called a THT. It's a kind of a dye that has a specific binding depending upon whether the protein is aggregated or not. It would not bind if the protein is monomeric state. It would bind if it forms some sort of an aggregate. Okay, so basically this dye is also fluorescent. So the fluorescent signal depends on whether it is bound or unbound. Okay, so you take this as a more like an assay, a fluorescence assay, and then we can see we look at the fluorescence coming out of this dye molecule as a function of time. When we put this dye molecule in the presence of a protein, and if the protein tend to aggregate, you will see an enhanced signal like this. Okay, so here you have a monomeric state, the signal is weak, but then if this aggregates form, if this monomer forms some aggregates and then form fibrils, and then this dye would actually more specifically bind to this, and you get an enhanced fluorescent signal, and you can see this sort of a sigmoidal kind of a curve. And you can detect the kinetics by looking at this simple approach. Now, we were actually looking at this problem from multiple aspects. We want to understand what is the mechanism of this, amyloid beta aggregation, and how do we interfere or how do we prevent this aggregation through some sort of additives, and then also how do we actually disintegrate or disassemble these aggregates even after they form, whether this is a reversible or not. And we are also interested to know whether these aggregates are actually affected by the flow conditions, the shearing effects and other fluid mechanical aspects. And finally, we were also interested in looking at how do this, there are a lot of biopolymers in our body, and that can also interfere with the amyloid beta aggregation. So what does that affect? So these are a set of questions that we've been looking at in the last 5-6 years or so. Maybe I will quickly run through some of the previous work, and then I'll go to the LLPS part. So what you see here is this is a TEM micrograph of those fibrils. You see this thread-like objects. Those are the amyloid beta fibrils, and these are the based on the fluorescence assay, you can kinetically study. You can study the kinetics, as a function of time, how much fibrils is formed. So what you see, this is the control. That means only protein is allowed to aggregate, and you see all the monomer goes to a complete aggregated state. Now, if you want to prevent this aggregation, what you could do is you can actually treat them with some nanoparticles, chosen with respect to the size and charge and so on. And depending upon how much nanoparticle you put, you can actually arrest or prevent the formation of aggregates. Okay, so we know some mechanism by which we can interfere the course of kinetics. On the other hand, if you start with a fully formed fibril like this, and you want to say how do we disassemble this? So in this case, these fibrils are quite stable, and they will not disassemble by itself due to some small temperature pH changes. What we need to do is we put some plasmonic nanoparticles like gold nanorods, and then we shine with some NIR light, and what it creates is some local hotspot, and this allows this protein to disassemble and disaggregate. And you see that as a function of time, these protein basically disassembles and disassociates. So these two results basically say that on one hand, if you start with the monomer, and if you want to prevent aggregation, you can treat them with some medicines, like nanoparticle or other things, to prevent the aggregation. That's one learning. On the other hand, suppose if these aggregates are already formed, and you want to look at a way that you can, get the monomers back. So you have to treat with the different kinds of particle and give some thermal, energy, and you can also kind of bring it back. So those are some of the things that we learned from this work. And we also looked at the effect of shearing and the effect of mixing, and how these processes affect the aggregation. This is because these monomers can be carried over through our blood vessels, and blood as it flows also subjected to a lot of shear. In the conventional context, people have studied that if you shear an aggregating system, a shear would actually prevent aggregation. Okay, so that's what people have shown before. But in this protein, we found an opposite effect, that if you do a shear effect, it actually kind of does a different role. So here on the first hand, we have a simple co-ed cell, and we apply a uniform shear. In this case, you can see that the control, if you don't do anything, if you just put a quiescent liquid, over a period of 40 hours, there is no much growth. But then if you kind of apply shear, then you actually see that the rate of formation of aggregate gets faster. Okay, so here we, sorry?
This is known and reported. It depends on the mechanism. So when the shear causes the developing aggregate to break up, then you have many more seeds. And so you have much faster kinetics.
Secondary nucleation, So what I was trying to say is that many of the globular protein aggregation, when people look at study shear effects, it used to be a breaking event, mostly breaking event. But here on the other hand, we have this kind of a secondary nucleation phenomena happening to facilitate faster nucleation. On the other hand, if you do a simple irregular mixing, which kind of gives more collision probability between the monomers, then you see a faster rate of formation. So we understand the aggregation kinetics from all these aspects. Now coming to this effect of biopolymers on aggregation, we were interested in on this polyphosphate. Polyphosphate is actually implied on many important biophysical systems, like it is involved in cell signaling, in sort of cell death, inflammation, and even metabolism of energy and so on. So, okay, so it's a very kind of a common workhorse in our body. Polyphosphate does many such work, and we were interested to see how these polymers might interfere with the polyphosphate, again from an in vitro point of view. So here we did this experiment with two different conditions. This is a neutral pH, and the other one is an acidic pH. So when we say acidic pH, we could be like 4.5 or maybe 3, which is very highly acidic, and the outcomes are very dependent on the pH. Okay, so when you are looking at neutral pH, when we add the polyphosphate of different amounts, it basically suppresses the aggregation rate, just like how the nanoparticles were acting on. Okay, so it has a more like a nanoparticle like an effect to prevent the aggregation rate. On the other hand, when we look at this acidic pH, in this case, the polyphosphates actually seem to enhance the rate of aggregations. So the black one that you see here, that's a control. As you increase the concentration of polyphosphate, you see that the aggregation completed much faster. Okay, so we see that the aggregation rate increases with the polyphosphate under neutral pH. Basically, it basically means that polyphosphate promotes aggregation of amyloid beta. This is very significant because when we look at cancer or many other cell inflammation, people have found that the pH in those cells are much acidic. Okay, and the biophysical process happening in an acidic environment can be quite different. So we see that in a normal condition, the polyphosphate are not really enhancing the aggregation, but rather it actually suppresses. But then suppose if there is any pH fluctuation in the cell leading to an acidic behavior, you can actually then do the opposite effect, it seems. Okay, so that's the main takeaway from this aggregation studies. And now we actually go further to see what is the nature of aggregate formation in these systems. Now, if you look at the the polyphosphate effect, I hope you can see here. This particular diagram is basically optical microscopic images of the system. We have treated at different. This is at acidic pH. So you can look at that. We have added 40 micromolar, 80 or 200 micromolar for a fixed concentration of the protein, and you see the development of droplets as a function of time. So this droplet formation is a significance. I mean, it just basically signifies the liquid-liquid phase separation in the system. And what you can also do is that in the system you can also study turbidity. And the development of turbidity also tells you that there is some growth of droplets. Yeah.
Without the polyphosphate, there won't be.
Yeah, no. So if you see here, this pH 4.5 at control, you see that it forms aggregate, but you won't see a droplet. Okay, so it will be just an empty image. I mean, you can't see anything. But if you do TEM, transmission micron electroscope, you will see a fibril structure that I showed in the beginning. You can see that just like how the THT fluorescence assay increased, in the same way the turbidity also increases, indicating the formation of this droplets and two-phase initiation of two-phase separation, phase separation. And again, we are showing that the effect of increasing the concentration of polyphosphates. So this is 40 micromolar, this is 80 micromolar, this is 200 micromolar, increasing in the concentration. On the other hand, when I put 200 micromolar of polyphosphate at neutral pH, there is no turbidity. It's a clear solution. No droplets, no aggregates phase separating. And then we can also do further analysis by looking at confocal microscopy. We can dye the system and we can see the droplets forming here. And we also did fluorescence quenching experiment, fluorescence bleaching experiment to see whether the droplet that we see are actually liquid or not. Okay, so if it is a liquid, if you bleach a spot, it will regain the fluorescence again. It's a very typical experiment to be done to characterize the liquid-like nature of the droplet. So we take a spot, we first bleach it, and then we wait for it, and then it regains the fluorescence. It kind of indicates there is a very diffusive transport indicating liquid-like behavior. And then we can also construct this kind of phase diagram between the concentration of polyphosphate versus the concentration of protein. And you see that one region where it does not form any LLPS, you need a critical concentration of polyphosphate to induce this. The one on the right hand, the green region here, that's where you see phase separation, LLPS. Whereas at low concentration, you don't see. So there is always, for a given amount of protein, you always need a critical amount of polyphase to induce the phase separation here.
Is this 80 or
40. 42 also we have done, but I have not shown it here. It does the same way. In fact, 42 is more prone to aggregate. So yeah. And then we can do further. Okay. Yeah.
I have just one question.
Sure.
So, is it just polyphosphate or any anionic chain can do it? Because there are like sulfated sugars like GAGs.
Yeah.
So, if they also can exert same effect or?
Yeah, so I will explain to you later that this effect is coming because of electrostatic interactions between the polyphosphate and protein. As long as the biopolymer that you add has a negative charge, then it should work. Okay, so yeah. So there's nothing specific about polyphosphate other than the charge here. The biopolymer you mentioned, is it charged?
So, like sulfated sugars.
Sulfated sugars, yeah.
Like, glycosaminoglycans.
Okay. We have not tried that, but the principle we are looking at is typically electrostatic driven. So as long as the biopolymer is negatively charged, it should work. Yeah. Yeah. Okay. So so then we further looked into the aggregate formation. So if you see at pH 7, you see that you get this kind of a fibrillar network. This is a normal case. But then if you increase the concentration of polyphosphate, you don't get any fibrils anymore. You get a little amorphous aggregate, which is different from the fibrillar network that you see here. On the other hand, when we look at this acidic pH leading to LLPS, and then if you look at these droplets, if you dry the droplet and you look at inside, we do see fibrils inside those droplets. So what we are saying is there's a two-step process. In the acidic condition, when we treat polyphosphate and protein, the first stage is to form the liquid droplet, and then within the droplet, the protein concentration is enriched, and that leads to an aggregation as if it would happen in a bulk system. So you have a liquid-liquid phase separation followed by liquid-solid phase separation, or crystal growth in other words. So that's what you see, that inside these droplets you see large number of fibrils that are formed. So we say that the fibril formation is actually amplified, enhanced due to the formation of liquid-liquid phase separation. Yeah, people have done it, but we have not done it. People have done it. Actually, in fact, if you have LLPS, compared to the average background concentration, the concentration inside the droplet can be 100x. In other experiments, people have measured. We have not measured though.
is sort of in a droplet versus bulk?
Yeah. So
Same morphology?
we see a little rough fibers in the LLPS compared to the bulk experiment, but looking at fiber level structural information is a little tricky. You have to go beyond TM. You have to do some people have done AFM kind of a study to see the local pitch is different, local structure is different. But from this same analysis, we found that these are a little thicker and rougher compared to the bulk fibrils. All right. Okay. So basically, I think the key message I want to show here is this effect of pH and polyphosphate in the aggregation behavior of amyloid. I started 5 minutes a little late. I hope I'm not. Okay. Okay. Just 2-3 more slides. Yeah. I know that all of you are hungry. Always, the talk just before the lunch is always the worst time to give a talk. So, anyways, I'm hungry as much as you are. So, if you look at the amyloid beta, cartoonistically we have shown like this, a monomer system, and this is a polyphosphate. When you have amyloid beta polyphosphate at neutral pH 7, the overall charge in the amyloid beta is still negative, but there is a region in the protein that is prone to aggregate, okay, and those regions have actually positive charges. There are a couple of residues which have positive charges, and due to the electrostatic interaction between the negatively charged polyphosphate and the positively charged local aggregating region, that basically leads to the prevention of aggregation, prevention of amyloid fibration. So that is the hypothesis we are making here. On the other hand, when you look at acidic, the entire amyloid beta, there is a lot of positive charge groups, and then again, the polyphosphate still remains negative regardless of the pH. In this case, we are talking about like an oppositely charged mixture of polymers. That we know that if you have oppositely charged mixture of polymers, they would phase separate through electrostatic coercion, and that's what you see, that the leading to the formation of droplets, and within the droplets, due to the enhanced concentration of the fibril proteins, you end up getting fibrils. So that's a kind of mechanism we have, and the way to test this, I'll probably skip this secondary analysis. And the way we can test this analysis is that we can do a molecular dynamic simulations. So again, this is a 40 residue long time scale experiment. So what we did is we took a small region where they will aggregate, and then we treated with the polyphosphate, and we looked at the how much amount of beta sheets and alpha helix and other structures are there between the two pH. And this is an initial results. We are not seeing the complete story here, and there seems to be some suggestion that from the simulation that there is a more beta sheet formation in the pH 3 compared to pH 7, even at a single monomer and polyphosphate level. But then going forward, we want to look at a more Goldstein model to see a phase separation in the full scale system. This also is skipped. The one last point I want to make here is that so far all of us have been working on DNA, amyloid, and RNA, those kind of biologically, human-related proteins. But there is a whole lot of proteins out there which are intrinsically disordered, but are there in the plant world. So what I'm showing here is a seed, a cross-section of a corn seed. And in the corn seed, you see there is this the dotted region here. Those are the starch and gluten. And if you do a SEM analysis, you will see that there are a lot of small droplets that you see here that are shown by the arrow. And these are nothing but protein droplets. And this has a lot of resemblance to the membraneless organelles that we showed in the in the animal cell before. So we are actually interested to look at how these protein droplets are forming in the seed, and this is something one can see in variety of seed proteins. Okay, there's also a nice book on seed protein to talk, that kind of tells you what is the composition of seeds. So I want to draw your attention that while one can do DNA, RNA, and proteins relevant to our health, human health, but there is also equivalent set of problems to be studied in the plant world also, and we hope that we can make a lot of useful study in those domain as well. So with that, I thank you all, and I'll be happy to take your questions.